Okay, so I should be online. Can people hear me? I can see people in the audience. Can you hear me? Not yet?
Very well.
Can you hear me? Oh, okay, I see somebody. Okay, there you go. Okay.
Yes, we can hear you.
Great, terrific. Thank you. So I'd like to start by thanking the organizers and in particular Thea Tilsty for helping to arrange my participation in this conference. I've been listening with great interest to the earlier talks in this session, but I wasn't able to attend the morning presentations, and nor will I be able to participate tomorrow. But I'm very happy to tell you a little bit about some of the work that we've been doing here in my lab and through various collaborations over the past number of years. Let me just give you a little bit of a sense. If Aisha, could you move it on to the next slide, please? Yeah, okay, so here's where I'm located in Calgary. It's a nice view of our city and with the Rocky Mountains behind us. And you can think of us a little bit like Denver, Colorado North. A little bit chillier perhaps, but still very pretty. Before I was here at the University of Calgary, long ago, I was a graduate student and then a postdoctoral fellow at Stanford University. And the postdoc in particular was with Bob Shimke, and I'm sure Thea Tilsty would have already told you about some of our joint adventures while we were postdocs together at the same time in the early 1980s. So what I'm doing now, next slide please, Aisha. Mainly in my lab we're looking at the use of oncolytic viruses to treat various kinds of cancer cells in vitro and in vivo, and we're looking at two main viruses in my lab, reovirus and also an engineered Coxsackie virus. And you see a couple of pictures here that show how we can have xenotransplanted human tumors in various kinds of nude mice and skid mice, and when we give them an injection with either reovirus on the left few panels or with Coxsackie virus on the right panels, with fibrosarcoma or with various types of gastric cancers, within about 8 to 10 days, what we find is that the tumors shrink away, disappear, and the mice are cured. And so reovirus is currently in clinical trials, phase 3 clinical trials in fact, and we've got a company that my colleagues here established at the here in Calgary that's sponsoring those trials, and they're going very well, and we're hoping for FDA and Health Canada approval for clinical use later this year or early next. And the engineered virus we think is going to be even better. So if we combine the effects of those two viruses, it looks like they can accommodate somewhere between 60 and 80% of human cancers can be killed by these viruses. And it's a completely different talk, but the mechanisms by which these viruses kill the cancers is by induced apoptosis, and it's responsive to various signaling pathways, including the activity of RAS signaling and perhaps, as well as we recently shown, tumor suppressor pathways as well. Now, I show this not because it's the main focus of my presentation, but rather to set up the real question that I'm going to talk about today, which is, why do some of these viruses kill some cancer cells, or chemotherapy or radiation therapy? Some cancers are highly susceptible to these therapies, some are not very susceptible. Each of the different types of cancers has different phenotypes as they grow within the patient, and so there's a huge amount of biological variability. So where is that all coming from, and how can we use that information to better understand the disease and diagnose it, and especially to treat it? And in particular, what are the genetic and genomic origins and consequences of these changes? So Aisha, the next slide please. So these are the main questions that we're looking at. How do cancer cells differ from normal cells, the origin, and can we use this knowledge in a constructive way? Next slide, please. And the story that I'm going to tell, and possibly Thea might have mentioned this earlier, but it really begins roughly a century ago when Theodore Boveri was the first fellow to contemplate the genetic and genomic origins of cancer, and he was one of the first people to begin to accept the idea of the chromosomal theory of inheritance. and to notice that chromosomes differed when you compared them from one species to the next, and in particular that chromosomes also varied significantly when you compared cancer cells from a patient with cancer compared with the originating cells. And so he had proposed roughly 100 years ago that chromosomal abnormalities in structure might be associated with differences in content and the passage of genetic information from one generation of cell to the next, just as they are also presumably associated with the passage of information from one generation to the next. So it was an intriguing idea, quite radical at the time, and of course in the absence of any genomic technologies it was very difficult to test. But as time went on, people developed those technologies, and one of the advances that really led to a much better understanding of the kinds of genomic variability that exist within cancer cells began in Shimke's lab when he had a visiting scientist, Joe Bertino. Aisha, next slide please. And Bertino and others were interested in the question of how cancer cells can acquire resistance to chemotherapeutic drugs, and certainly this is a grave limitation to the use of chemotherapy with patients. You want to adjust the dose so that it's high enough that ideally you kill the cancer cells without quite killing the patient, and sometimes that works and sometimes it doesn't. Sometimes the tumor goes away for a while, then comes back and appears to have been altered so that it now can grow even in the presence of drug concentrations that previously would have almost killed the tumor. And when you look at the chromosomal characteristics of those cells, the newly drug-resistant cells, what one finds is that there are often chromosomal abnormalities, and those are of two main varieties. One can have extrachromosomal elements, often referred to as double minute chromosomes, and one can also have expanded regions within the chromosomes that have abnormal GIMSA G-banding staining patterns that are referred to as homogeneously staining regions. And to make a very long story short, because this has been well documented in the literature, and Shimke's lab was coming up with these observations just at about the time, or a year or two before, Thea and I had both joined his lab as postdoctoral fellows. The bottom line is that what was happening is that particular genes were varying in their copy number, and in particular increasing. Those genes encoded transcripts and then proteins that were associated with the chemotherapeutic drugs in question. So, for example, methotrexate, which inhibits the enzyme dihydrofolate reductase and thereby blocks metabolism, one-carbon metabolism, which is required for thymidine synthesis and therefore DNA replication, particularly in rapidly dividing cancer cells. So by amplifying the number of genes encoding that particular enzyme, more enzyme could be produced and cells could survive in the presence of high concentrations of drug that would otherwise kill normal healthy cells. So next slide, Aisha. One of the questions that I was working on in the 1980s in Bob's lab was whether that kind of variation might arise spontaneously within cells in the absence of any particular selection pressure. So we came up with a method relying upon flow cytometry, and we actually used the very first flow cytometer that was ever developed in Len Herzenberg's lab at Stanford, and we were passing cells through and staining them with fluorescinated methotrexate to look for cells that had variations in the quantity of the enzyme under conditions that were non-selective. So there was no metabolic selection pressure on them whatsoever. We were compensating for the inhibition of the enzyme, and everything seemed to be working out quite well. And we would select for cells that had higher or lower levels of fluorescence intensity, therefore higher or lower levels of enzyme content. And then when we looked at how quickly that was happening, and here on the left side of the screen I'm showing a southern blot with varying levels of intensity of hybridization to radioactive dihydrofolate reductase probe and various control specimens. And in this case I was using Chinese hamster ovary cells that amplify their genes not by double minute chromosomes as was shown on the previous slide. Those instead the Chinese hamster cells amplify by homogeneously staining regions, so tandem duplications within the chromosomes. And what we found was that even in the absence of any metabolic stress or mutational pressure or anything that we could monitor that indicated that the cells were under selection, aside from selecting the rare variants that arose spontaneously that happened to have more or less enzyme, they also had greater or fewer numbers of copies of the dihydrofolate reductase gene. So we went through our numbers and calculated that in fact the frequency of appearance of these spontaneous events of amplification or deletion was astonishingly high. So at this particular locus, in the absence of any mutagenic pressure, what we were finding was that we were obtaining events of amplification or deletion at rates as high as 10 to the minus 3 events at this locus, not genome-wide, just at this locus per cell division. Subsequently, we then went on to look at, so if it's happening spontaneously that frequently in these Chinese hamster ovary transformed cells, what would happen if we then expose them to a variety of chemotherapeutic agents? And just as Tom Glover had been speaking about earlier, so this was a paper that we published in, I think it was 1985 or 86, something like that. And this was after I had left Shimke's lab, but rising from work that we had begun there. So we exposed the cells to hydroxyurea and afeticolin and other types of agents, and methotrexate, of course, agents that can interrupt progression through the cell cycle by interfering with DNA synthesis. And we would do this in a transient exposure to the inhibitory agent. Cells would block, pile up in the cell cycle, then we would release them from that inhibition, and what we would find was massive increases during that release, massive increases in total DNA content, and a further remarkable increase in incidence of amplification events of many, many different genes, including of, let's say, dihydrofolate reductase. And so we estimated from those studies that we could get a tenfold at least further increase in the frequency of occurrence of these amplification events following transient exposure to these agents that would interrupt cell cycle progression. Independently, Thea, in her experiments, and perhaps she spoke about this briefly this morning, had shown that exposure of cells to specific DNA damaging agents also promoted an increase in the occurrence of genome amplification events. And then Thea Tilsty and Jeff Wall independently subsequently went on to show in a series of very, very nice papers that those really high frequencies of genome amplification and deletion happen preferentially in transformed cells that show deletion or inactivation of DNA damage response pathways, so for example P53 and retinoblastoma and so on. So conclusions that we were beginning to draw so many years ago were that amplification events could occur spontaneously with surprisingly high frequency, particularly in transformed cells, and that the rate of occurrence would increase further when DNA damage occurred or interruptions in cell cycle progression. Next slide, please. So if that is true, and here I'm showing a picture of one of the double minute chromosomes that we had taken in a collaboration with Barbara Hamcallo. So the amplification events can occur in the extrachromosomal structures, which contain origins of replication. The extrachromosomal elements map as circular genome elements. In addition to an origin of replication, they have the gene of interest, but no centromeric DNA, no telomeric DNA, and they can vary extremely rapidly in copy number by random segregation among daughter cells at mitosis. But we're also getting those big changes occurring in the homogeneously staining regions as well. And in subsequent work that we did in my lab and in many other labs around the world, we showed that the amplification occurs not simply in genes encoding enzymes involved in responses to chemotherapeutic drugs, but also cellular proto-oncogenes, so c-Myc and n-Myc, that deleted genes include the tumor suppressor genes, that these changes can be passed in heritable fashion from one generation to the next and are associated with Li-Fraumeni syndrome, etc. So lots of those things were being demonstrated in my lab and in other labs at that time. We also showed that as copy number varies, we can observe effects in trans on other genes. So we specifically varied the copy number of promoter regions of genes and showed that we could titrate out transcription factors. We did this for heat shock genes, for c-Myc, and for other metallothionein genes as well. And so we could up or down regulate whole families of genes by titrating out transcription factors in vivo in cells and regulating the genes up or down depending on whether we were titrating a positive or negative acting transcription factor. So this was a very interesting series of observations that were associated with the cancer cells. And of course, we've heard about this elsewhere, and Jennifer mentioned this as well in her presentation, and Joe touched on it briefly. So a lot of copy number variation exists naturally within the human genome, and so the slide that we're seeing up here is taken not from our work, but from a paper that was published 2 years ago in Nature, looking at copy number variation that exists naturally within the human population. And of course we see, if we look at comparing different species within the mammalian lineage, or even going back much further in chordate ancestry, huge variations in copy number that exist within germline cells. But if we look across many different types of cancers and try, so Aisha, next slide please. So here we're looking at many different carcinomas, doing CGH array measurements of copy number variation. Again, another paper that was also published 2 years ago in Nature from a different group, but we've got our own data that I'll be showing you in just a minute. So we can see regions of genome amplification. So on the vertical axis, we've got chromosomes 1, 2, 3, 4, all the way down to the bottom, chromosomes X, and so on. And on the right-hand side, genome amplification events that exist among many, many different carcinomas, all grouped together, and genome deletion events on the left side of the panel. And if you try to imagine, first of all, how frequent are these, and so the length of protrusion of the red or blue lines indicates relative frequency of these events, and so the greater the protrusion, you see frequent amplification or deletion events at that particular site within the genome. Small protrusions, they're relatively rare. It also gives you an indication of the size of these events. So one can think of focal amplification or deletion events, or events that encompass much wider regions of the chromosomes that can include dozens or even hundreds of genes within the domain that is amplified or deleted. In some cases, the amplification or deletion event is small in magnitude. In other words, you can go from a copy number of 2, a diploid content, up to a triploid or tetraploid content, not the 50 or 100 or 1,000-fold that we see in drug resistance associated amplification. But even so, and as Jennifer was mentioning in her presentation, as you observe variations in gene copy number, even 2 or 3-fold or a 50% decrease in copy number, one also observes changes in gene expression. So when most people look at these genome amplification or deletion events, they're often very interested in, oh, where's the c-Myc gene, where's the p53 gene? Is that sufficient to explain these large, either the focal amplification or deletion events or the larger ones that we observe? So I understand the focus on the contributions of individual genes, many of which are shown here, that can be gained or lost in copy number. But Aisha, if you can go to the next slide, please. So this is some of the data that we're generating in association with our collaborator, Michael Bautis. He's in Switzerland, and he's doing his own
Dr. Johnston? You're... Is he still there?
Oh, okay. Are we back? Looks like we are. Okay, there was some strange interruption there. Okay, so we've got a collaborator in Switzerland, Michael Baudes, with whom we've been collecting these data. So here is shown another representation of data looking at amplification and deletion events. And so here the height or the depressions, so the amplification events are shown in green across the entire human genome, and deletion events are shown in that orange-red color. So what we're looking at here is the frequencies of these events. So up to 30% of all human cancers, for example, will show amplification of the long arm of chromosome 8 where the c-Myc gene is localized, and 10 or 20% will show deletions of chromosome 8p, etc. The point I want to make from this, in part, is look how broad are the regions that can often show simultaneous increases in copy number or deletions. So these are very, very large chromosomal domains, often associated with telomeric DNA, but stretching in many cases all the way back to the centromeric regions. Aisha, next slide, please. So we began to think about what on earth are going on with these very large domains. And Jennifer had touched on this briefly about the nature of our genomic organization, and the mammalian lineage, if you compare us with our earliest chordate ancestors, so let's say the cephalochordates like Amphioxus, our genomes, you could say, are approximately pseudo-octoploid. So there were two whole genome duplications that occurred early during chordate evolution. On occasion, additional genome duplication events have occurred. So teleost fish went through another genome duplication. Amphibians independently went through another one. So those guys are hexadecaploid. So duplication events and then selective gene loss that occurs at various periods of time. Here is shown our own representation of how we think the remnants of those two whole genome duplication events are distributed within the human genome. So on the right side of this panel, we're showing the human genome, chromosomes 1, 2, 3, 4, all the way down to the X and Y chromosomes. And then on the left, distributions in certain clusters of genes where you see in each case, 1, 2, 3, 4 green or purple or orange or red, etc. clusters. So those are the ancestral domains that became subject to the quadruplication events, and then gradual reduction because some genes weren't always needed in 2 or 4 copies. So now that's why I say we are pseudo-octoploid. And then on the right we see how those are distributed among our various genes and families of genes within the chromosomes. So when you look at what are the regions that are gained or lost in human cancers, and also are gained and lost during evolution, so we'll touch on that in just a minute, what we find is that there are some remarkable patterns that most people seem not to have paid attention to. So these are our drawings that we're showing here. Independently, a couple of other groups have come across similar findings, but they haven't related this in any way to cancer cell formation. So Nakatani had a paper, and Putnam and colleagues have published similar reconstructions of ancestral genomes, but the way that we're taking it goes a little bit differently. So Aisha, next slide please. So if we look at one of these clusters of the quadruplicated regions and look at how those regions have been conserved during evolution over 300 million years, or probably a little bit longer, of chordate evolution. So all the way, chondrichthyes and fish and amphibians and going up through the theropods and finally the mammals and primates. Here we're looking at the distribution of one of these clusters. We call it the clusters are called parsomes, and we call this one the EGFR parsome, but we're thinking of changing the name for other reasons. Anyway, what we find is that these genes are partially duplicated within the parsomes. These are the 4 chromologs that were duplicated long, long ago and which we have retained many elements. They've been fragmented a little bit within our genome, but what's astonishing is how well they have been preserved and how many of the genes have been retained over 300 to 400 million years of evolution. And some of those genes are shown on the right-hand side of the panel, and they're associated with many, the proteins that they encode are associated with many regulatory events governing cell proliferation, responses to extracellular signals, cell cycle control, cellular differentiation, and all of that. And each of those different 6 parsomal assemblies that I was showing on the previous slide is associated with different families of genes that segregate together during evolution and that appear to play important roles in governing cell proliferation, cell signaling, cell differentiation, and the formation of the embryonic axis. So, the next slide shows if we do comparative analyses among different species. So the big circle in the upper left shows all of those colored domains for the parsomal structures as they are arrayed in the human chromosome, and then we show it kind of teased out for the different parsomes, the purple, the yellowy orange, the blue ones, the EGFR par that I was talking about, the green ones, etc. And you can see, yes, there's been a certain amount of fragmentation, but surprisingly little over 300 million years of evolution. And what we're mapping out with all of those swooping curves is how the genes that have homologs that are duplicated or quadruplicated because of the whole genome duplication events are present, distributed throughout the genome. So as you go through the chromosomes 1, 2, 3, 4, all the way up to 22, X and Y, what you see, of course, is regions where the certain colors are concentrated. Right in the very middle of the screen, you can see how well those are clustered among these conserved parsomes. Single colors, large regions that are evolutionarily conserved. And then along the lower panel, we see different species, so dog and mouse and so on. So each species has its own unique array, and yet the point being, these evolutionarily conserved clusters of genes, partially quadruplicated, show patterns of inheritance that are remarkably well conserved. So one has to ask, why are they conserved over such a long period of time? What are the evolutionary selection processes that keep large arrays of genes in close proximity? And what happens when they start to undergo redistribution, changes in copy number, etc., as cancer cells begin to appear? So, next slide, Aisha. So now we're looking at the distribution of cell cycle and proliferation-related genes among these various parsomes. And so we see various cyclin and CDK genes that are distributed in this way. All kinds of different genes can be distributed among the various parsomes. They can be gained or lost in the cancer cells. The next slide, please. Mitogen activated protein kinases, so MAPK associated genes. Again, we see particular conservations among these. So the tyrosine kinase receptor associated genes in the plasma membrane are associated with the EGFR par. Some of the regulatory components are associated with some of the other parsomes. So we have a distribution of function among these conserved elements. And next slide please, when we look at the regions that are frequently gained or lost among the human chromosomes, so here we show again the human genome but arrayed in a slightly different way. we're looking at how frequently either entire chromosomes or regions of chromosomes on the P arm, the Q arm, are gained or lost. Some chromosomes tend in cancer cells to increase in copy number. That's on the left side of the left panel. The next one over, showing the red, shows those chromosomes that are frequently lost in human cancer cells. The green ones show chromosomes that, sometimes they're lost, sometimes they're gained. It's a bit variable. And then on the right part of that left panel, we show some chromosomes, chromosome 3 and 8 and so on, that show large variations. Half of it is gained very frequently and half of it is lost. And when you look at the genes that are present within these gained or lost regions, you can reconstruct signaling pathways that govern proliferation within cells. So look at the lower right side of the panel, and what you see are, shown in blue, are genes contained within chromosomal domains that are frequently increased in copy number in many different carcinomas. So the EGFR, EGF receptor gene, cyclin B, cyclin E, CDK2, etc. And you also see in red genes encoding proteins that are frequently lost. So the retinoblastoma and other genes that inhibit cell cycle progression and DNA damage responses and all of that. So what we're finding, therefore, is that as we gain or lose copy number, it's not just individual genes, and we've got a dozen of these little charts showing different pathways, showing gain or loss of cell cycle control genes, response pathway genes to extracellular signals, differentiation genes, tissue-specific genes that might be gained or lost. It's really showing a pattern. So it's not just individual genes when they're increased or decreased in copy number. It's actually dozens or hundreds of genes that we think are contributing to the cancer phenotype. Then we asked a very interesting question, and we're getting some quite surprising results. So the next slide, please, Aisha. So here, what we're finding is, and again, we're building on the data that we obtained from our collaborator, Michael Bautis. So he's got CGH array data for all kinds of different cancers, and we've been collaborating in the detailed analysis of this. So we're looking here at gains or losses in colorectal cancer, and we've got similar chart. I'll show you breast cancer in a minute. We've got brain cancers and leukemias and lymphomas and prostate cancers and lots of different ones. What we're doing here, and I don't think anyone has ever come up with this kind of a display before, we're looking at codependent variation in copy number. So let's just look at the left panel in colorectal cancer gain or loss. Along the bottom, you can see half of one of those Bautis histograms showing frequencies of gene amplification events in colorectal cancer arrayed across the human genome. Chromosome 1 on the left, 2, 3, 4, all the way up to 22 in X and Y. And what you see is in colorectal cancer, there are certain frequencies of gain or loss, and again, for example, human chromosome 7 is frequently gained, chromosome 8, 8q is frequently gained, etc. Then we say in those, and he's got roughly 800 or 900, over 900 now, different tumors from individual patients that are shown on these arrays. And we're saying in each individual patient, if that patient has, let's say, a gain in human chromosome 8q in that large domain that contains hundreds of genes, what is the likelihood that they will also show a gain or a loss elsewhere within the genome? So as you go along the horizontal axis at the bottom, you can track on to chromosome 8q, then look upwards, and you see green regions where there are domains that are often gained only when 8q is gained. Red regions, those are often lost. Then other regions, green, they're gained, red, lost, etc., etc. And similarly, when you look at the right-hand side of the panel, because it's an independent analysis, when you look at regions that are lost in human colorectal cancer, and then you start marching up from those regions, so 8p is often lost in colorectal cancer, and you go up from there. So human chromosome 1 is not frequently lost when 8p is, but there are regions in human chromosome 2 that frequently are lost. So we've got a co-distribution of gains or losses, codependent correlations among these events. So that if you look on the vertical right-hand side of each of those panels, what you see is the distribution of those evolutionarily conserved chromosomal domains that correspond to those ancient duplicated regions. And what we find is that the regions that are frequently gained or lost correspond with those chromosomal conserved domains. Next slide, please, Aisha. And so here, I won't go through it in detail because I'm already running out of time. So we're finding similar kinds of gains or losses in breast cancer and prostate and lung, different patterns. Each type of cancer has its own unique spectrum of gains and losses. There are some similarities. All carcinomas show some patterns in common. Neuroectodermal, so glioblastomas show their own unique patterns. Leukemias and lymphomas, different again. Sarcomas, different again. So we're finding patterns that are associated with signaling pathways in those various types of tissues. Some are tissue-specific, some are tumor-specific, codependent gain or loss, and it's associated with the distribution of these evolutionarily conserved gene families. They're conserved in their topographic clustering within the genome, conserved in their function in controlling cell proliferation, cell signaling, cell differentiation. And so we're beginning to think that these copy number variations are not simply associated with the gain of c-Myc, the loss of p53. Those are certainly important, or mutational events, or gains or losses in regulatory RNA, but we're finding that it's entire families of genes that go up or down in copy number, twofold, threefold, fourfold, whatever it is, that are associated with changes in cellular phenotype. Next slide, please. And I'm almost wrapping it up here. So, the conclusions that we draw from our analyses and from many others, certainly aneuploidy is frequently observed in many different types of human cancer. So we have mutational events, epigenetic events leading to altered expression patterns of large sections of chromosomes, and also variations in copy number of large regions as well. So those changes can arise quickly. Copy number can change rapidly, especially in cells where genomic integrity has been lost by inactivation of p53 or RB or other DNA damage response genes. These can be important contributors to oncogenesis, to tumor heterogeneity, and to drug resistance, and many other aspects of the phenotype of individual cancer cells. The distribution, we conclude, of gene clusters in our genome is non-random, so we're recently undertaking much more detailed analyses of gains or losses and of evolutionary conservation, and we're finding some remarkable examples of conserved clustering, but local variability in topographic distribution. So micro explosions, you could say, almost within genome organization. Very, very interesting. We're trying to figure it out, and we're getting really valuable clues from those ENCODE papers published a couple of weeks ago as to why that might be the case, and that the boundaries of these regions that are gained or lost correspond remarkably well with the evolutionarily conserved parsomal boundaries, meaning that hundreds of conserved and coordinated genes are involved in these events in a codependent manner. So they're coordinated in at least 2 dimensions. We're now undertaking 3- and 4-dimensional analyses, looking not only at gains and losses in chromosomal distribution, but also in point mutations, or inactivations or activations of various genes, RAS activation, all of that stuff, and as well with genetic changes. So we think that this is a very interesting way to think about cancer, how it arises, what is its relationship with evolutionary variation, how species differ from one another, and we think that these analyses are going to lead to improved insights into the origins of cancer, provide us with new diagnostic tools, and perhaps as well therapeutic targets. So anyway, thanks very much for listening. The last slide shows some of the people in my lab and our collaborators, and in particular with Christoph Simpson. He's a bioinformatician who's helped us greatly in devising these new ways of looking at genomic organization and chromosomal gains or loss. Thank you very much, and I'm happy to take any questions.
Questions from the audience?
So I really appreciated your talk and have a comment and a question. The comment is that I urge you to take a look at the model of preservation of gene duplicates that was put forward in the paper that we released with the Daphnia genome, because I think it might be pertinent to your work. But my question falls along these clusters of genes that you're finding conserved, and it strikes me that there's an analogy to draw with development from that, in that we see similar clusterings of developmental genes, and I even noticed that one of your sets there was a Hox cluster. And in development, we know where we have very streamlined trans-regulation working. A few Hox genes are controlling a lot, whereas it really comes down to cis regulation and all this variation in regulatory regions that's driving diversification. It seems one way to maintain that streamlined transregulation would be through gene duplication.
I was intrigued as well by your comments on Daphnia genome organization and evolution and how quickly those things can change, etc., and the patterns of their change. The distribution, I mean, everybody's familiar with the highly conserved gene clusters in Hox, in the Hox arrays, and in globin arrays, etc., and their patterns of coordinated expression, tissue-specific and temporal expression that are highly conserved. We are arguing that it may well go beyond those individual gene clusters into these much larger arrays. And when you look carefully at the papers that were published as part of the ENCODE Galaxy that was published a couple of weeks ago, several of those papers talk about coordinated expression patterns over multi-megabase domains of the chromosomes. And what we're finding is the domains that are beginning to be identified, because that was still a very preliminary analysis, only looking at a small fraction of the human and mammalian genomes, we're finding they correspond very, very precisely to the parsomes that we're talking about. So we're beginning to think that the conservation, subject to these micro-explosions within topography within the genome, that coordinated expression, up or down regulation in a global way, and then of course each individual gene needs to turn on or off as it's needed, I think that is subject to selective pressures and retention as much as individual genes.
Okay, thank you very much. Can we have the panel members come up for a few minutes for general discussion?